Good afternoon, good evening, good morning, everyone. Welcome to the first Axcelead Global Seminar. Thank you very much for participating in this seminar today. My name is Kaz Aso, Head of Sales from Axcelead Drug Discovery Partners. I'm leading this seminar today. At the beginning, Yoshi Ikeura, our CEO, will give you a short message. Please, Yoshi.
Hi, everybody. I'm Yoshi Ikeura, CEO of the Axcelead Drug Discovery Partners. Thank you very much for joining our webinar. Today, we'd like to introduce our unique screening service to identify the promising hit compound by using our unique and proprietary 1.5 million compound libraries. As you know, in a drug discovery process, hit identification is an extremely important process. The quality of the hit compound will affect the future of the project. If you could identify a good hit compound, probably the rest of the drug discovery process would become much easier. So our services would cover the broad range of the target class, and also our service would cover the new approaches attracting attention, such as the protein degradation and RNA focusing, RNA targeting a small molecule, and so on. So stay tuned to find out why we Axcelead can identify the promising hit compound for you. So before diving into the main topics, we are going to have a little housekeeping information from the Kazaso.
Here is today's agenda. We have two speakers. First speaker is Yoshi Ikeura, CEO of Axcelead Drug Discovery Partners. He will talk about company overview. The next speaker is Tomohiro Kawamoto, who is the Senior Director of Discovery Biology. He will talk about our HTS capability and our HTS service. Let's get started. Please start the presentation, Yoshi.
Okay, thank you. Again, I'm Yoshi Ikeura, CEO of the Axcelead. I'm going to talk about the company overview. Please go to the next slide. So Axcelead is a solution provider in the drug discovery space from Japan. Our company was established in 2017 as a Takeda spin-out company and became fully independent from Takeda in 2019. Currently, we have 270 employees. Majority of them are experienced scientists having industrial research experience. And we have more than 170 clients, which include not only the Japanese pharma and ventures and academias, but also the US ventures and also the US big pharma too. Please go to the next slide. So this slide describes the highlight of the Axcelead. So as I mentioned, we are the spin-out from Takeda's. So we took over the Takeda's preclinical R&D capabilities, including a unique and proprietary drug discovery platform, which means we have experienced scientists, market leading infrastructure, and also it is noteworthy that we have access to the Takeda's legacy data and can use those information for our business. So our capability include the broad range of the functions in a drug discovery spaces, as described in the right-hand side. And also we have a wide variety of the strong partners, which enables us to access to the new frontier technologies. And so we can generate a candidate by ourselves by leveraging all of the strengths, and then we can provide the wide variety of the services in any of the drug discovery stages, starting from the early exploration studies, hit identification, and lead generation optimization to identify the candidate, followed by the IND enabling studies. Today, we are going to focus on the HTS service to identify the attractive hit compound. Yeah, that's all from myself.
Thank you very much. I will invite the next speaker, Tomohiro Kawamoto, please. Tomohiro Kawamoto will talk about the driving innovative drug discovery through an integrated high-throughput screening platform. Please start your presentation. Thank you.
Thank you all for joining us today. I'm Tomohiro Kawamoto. I'm Senior Director of Discovery Biology Group at Axcelead. I would like to talk about our platform for hit identification to drive innovative drug discovery, including new therapeutic modalities. This is the outline for today's talk. Firstly, I would like to talk about our key factors for hit identification. These are key factors for hit identification. HTS is a key methodology to find hit and lead compounds for drug discovery. Axcelead has large-scale library, cutting-edge facilities, a broad range of assay platform, and well-experienced researchers. So Axcelead can provide comprehensive HTS services through our integrated platform. This is Axcelead compound library. Axcelead molecule collection contains over 1.5 million compounds. This is the quality of our compound library. QED is an indicator of lead likeness. 90% of compounds have a lead likeness property. 6% of compounds were originally designed and synthesized. The features of our library are lead likeness and diversity. These are library sets for HTS. We have two types of libraries, diversity libraries and forecast libraries. Diversity libraries are established considering diversity and lead likeness. Single library consists of 129,000 compounds. Full library consists of 500,000 compounds. It contains 10 different compounds per one sample. There are multiple focused libraries, for example, libraries for target classes, macrocycle compounds, RNA-targeted drugs, covalent compounds, extended rule of 5 compounds, natural products, biologically annotated library. Biologically annotated library is available for phenotypic screening. It is useful for mode of action analysis. And we are also able to construct a focused library selected from all library by virtual screening. This is hit identification process in Axcelead. It includes multiple steps to discover high-quality hit compounds. Strategy planning, assay development, primary assay, HTS, and counter assay, and hit selection. After that, we can offer both hit expansion and profiling in the post-HTS prior to the lead generation. So Axcelead can offer high-quality hit compounds with sophisticated strategies. This is a track record of HTS campaigns. We have successfully completed 698 HTS campaigns for various target classes with high hit rates. Based on a lot of experience, we can offer optimal strategies. This is our strength, HTS platform. This is the target-based assay platform. It includes GPCRs, ion channels, transporters, enzymes, nuclear receptors, protein-protein interaction targets, and nucleic acids. Our assay platform covers a wide range of target classes. This is a kinase platform. We have successfully completed this campaign for 81 kinases, including non-protein kinases. These are full range of assays, for example, enzymatic assay and binding assay, and profiling, including cell-based assay and biophysical analysis. We have original selective kinase inhibitors for 120 kinases through our global kinase panel consisting of 300 kinases. We call these assets ASCIP, Axcelead Selective Kinase Inhibitor Profiler, and we can make a drug discovery plan using these assets. We can accelerate your drug discovery with our assets as an integrated drug discovery service. This is an GPCR platform. We have experience with these assays. This is an actual example of HTS GPR 39 positive allosteric modulators. We developed an assay system to monitor cyclin P with FLET system and conducted HTS campaign with our library. Finally, we found hit compounds. This compound selectively activated GS signal, and so we can support discovery of GPCR biased ligand with our platform. A rapid-fire mass system enables us to conduct label-free assay by directly monitoring products and substrates. This is an actual example of discovery enzyme inhibitors using rapid-fire mass system. The target is LDHB, lactose dehydrogenase B. We monitored both NADH and NAD with rapid-fire mass system and conducted HS campaign with our library. Finally, we found hit compounds. This is a representative hit compound. This is the first selective inhibitor of LDHB. We clarified the binding mode with X-ray crystallography. This compound bound to the allosteric site on LDHB. This result supports the selectivity for this compound. We also have capabilities on in vitro assays for profiling after HS campaign, mode of action analysis, kinetics, cell-based assay, and biophysical analysis for target compound interaction assay, SMS affinity selection mass spec, and SPR, ITC, and X-ray crystallography. So we can drive the structure-based drug discovery from a hit compounds. Next, I would like to explain our approach to drugging unknown targets. Axcelead has two different strategies: target-based drug discovery, TDD, and phenotypic drug discovery, PDD. Each strategy has pros and cons. For example, target-based drug discovery enables us to use a structure-based approach. However, hit compounds may not show efficacy in cell-based assay. It depends on the target. On the other hand, phenotypic drug discovery is a straightforward method to discover compounds with cellular activity. However, target decomposition and MOA analysis can be challenging. Axcelead has capabilities of DDD, TDD, and PDD, and provides the best solution. This is the track record of phenotypic screening. We have successfully completed more than 70 HS campaigns with phenotypic screening. This pie chart and table show the assay methods and usual screening library size. For example, 30% targets are from imaging system using the high content assay. We mainly use 100,000 compounds for HS campaign. Our platform covers a broad range of assay methods. Based on the out of experience, we can propose the best solution according to your needs. iPS technology are applicable not only for cell therapy, but also for drug discovery. We have a partnership with Fujifilm Cellular Dynamics. They can provide a lot of iPS differentiated cells. We develop an assay system using their iPS cells and conduct HS campaign. And this is an actual example of HTS using iPS differentiated microglia cells. We developed an assay system to monitor the endocytosis A beta in microglia cells using TREM2 mutant gene. As you can see, the endocytosis activity in TREM2 mutant is decreased compared with the wild type. This is a disease-relevant assay system. So we tried to find activators of endocytosis in TREM2 mutant microglia cells. We developed an assay system and conducted HS campaign with our library. Finally, we found hit compounds. This is a representative hit compounds. This compound activated A beta uptake in TREM2 mutant microglia cells selectively. We are now investigating the mechanical action for hit compounds. MOA analysis can be challenging for PDD. However, we have identified the targets based on the annotation for hit compounds. This is the outline to predict MOA for hit compounds. To clarify the MOA, we confirm the phenotype using different compounds with the same annotation. Next, we analyze the correlation between biochemical assay and cell-based assay. After that, we will validate the hypothesis using sRNA or CRISPR-Cas9 knockdown technologies. We have capabilities on target deconvolution for target unknown compounds. In the last slide, I explained the annotation analysis. There are two other strategies: chemical proteomics with the bait probe for direct target fishing, and fingerprinting based on the NGS data analysis. So we can offer comprehensive platform for target deconvolution and support drugging unknown targets. This is a summary of phenotypic drug discovery platform in Axcelead. Using high quality and attractive libraries, cutting-edge facilities, and comprehensive platform, we can drive phenotypic drug discovery for hit lead finding, drug repositioning, and target discovery. Next, I would like to talk about our capabilities on drugging undruggable targets. Protein degradation inducers such as bifunctional degraders and molecular glues have been reported. And immunomodulatory drugs, thalidomide and its derivative, have been reported, and their molecular glues that can induce the degradation of neon substrate. In addition, there are a lot of reports about RNA-targeted small molecules. Representative risdiplam was launched for the treatment of SMN as a splicing modulator. These new modalities have shed light on small molecule drug discovery. The potential targets for small molecules have been expanding, so we have constructed drug discovery platform for these new modalities. These are capabilities on targeted protein degradation. We have constructed a molecular glue library consisting of 6,000 compounds. Regarding this platform profiling, we have experience with these assays. For example, degradation assay using hybrid system, ternary complex formation, ubiquitination, and ASMS binding assay, and phenotypic screen. Actually, we have a track record of 16 programs for targeted protein degradation. This is an actual example of discovery of protein degradation inducers. In this case, we developed a cell-based assay using target hybrid knocking cells. The target protein fused with hybrid is detected by a luminescence assay system. This is a robust cell-based assay to detect endogenous expression of targets. We developed a system to conduct HS campaign using our library. Finally, we found hit compounds that can induce the degradation of the target protein selectively. These are capabilities on RNA-targeted drugs. We have constructed focused libraries. Using focused library and diversity library, we have conducted HS campaign with these assays. For example, RNA-BP binding assay, fluorescence proof binding, and ASMS binding assay, and cell-based reportage assay, artificial and other phenotypic screens. Actually, we have a track record of 15 programs for RNA-targeted drugs and one joint research with these companies. These are RNA-focused small molecule libraries. The first one is general RNA-focused library. It consists of 6,400 compounds selected from all library. This library was selected by multiple analysis of RNA binders, RNA co-structures information. Second one is RNA splicing focused library. It is an originally designed and synthesized compounds consisting of 1,700 compounds. The design concept is based on the interaction patterns between diverse splice sites and snRNP, so it is useful for discovery of RNA splicing modulator drugs. This is the actual example of assay result for our splicing focus library. qPCR was used to detect the RNA splicing of SMN2 genes and other target genes. Probes were designed based on the exon skipping region to monitor the RNA splicing. We summarized the assay result in the heat map. We found heat compounds affect RNA splicing as of SMN2 gene, as well as other target genes. And several compounds show the gene selectivity. So we can offer integrated drug discovery service using this forecast library. These are actual example of discovery of RNA binders with high-throughput ASMS. ASMS affinity selection mass spec is a biophysical binding screen. It enables us to conduct HTS for targets not amenable to traditional biochemical screening. We conducted HTS for FMN riboswitch RNA binders using diversity library and general RNA focus library by high-throughput ASMS. Finally, we found heat compounds. This is a representative compound. This compound bound to the FMN riboswitch RNA with KD value of 65 nanomolar. This compound competed with its natural ligand FMN. We clarified the binding mode is X-ray crystallography, so we can drive the RNA-targeted drug discovery with our platform. After completing this campaign, we propose our post-HTS and lead generation service with our medicinal chemistry team. Extensive information about newly synthesized compound from heat compounds and its SNR analysis enables smooth transition from heat identification to lead generation stage. Axcelead can accelerate lead generation from advanced heat compounds with efficient DMTS cycle. To accelerate this DMTS cycle, we use multiple in silico technologies. We call our original in silico tool ACO, Axcelead High Performance Computing System for drug design. So we can increase the efficiency of drug discovery based on a lot of experience and multiple in silico technologies. This multidimensional approach is required for drugging undruggable target with Axcelead's excellent chemistry team. I would like to summarize the points so far. We have three strengths. Firstly, we have pharma origin attractive libraries. Secondly, we have state-of-the-art infrastructures, including fully automated screening system. Thirdly, we can offer high quality and comprehensive services. They include strategy planning, assay development, HTS profile, and heat expansion, and other methods profile. So we efficiently offer high quality hit and lead compounds through our integrated HTS platform. Axcelead can accelerate your drug discovery. That's all for my presentation. Thank you so much.
Okay, thank you very much. Your presentation, Tomohiro. I received many questions from the audience. So let's move to the Q&A sessions. Okay, the first question is about the time frame of our HTS service. How long does it take to conduct the HTS campaign until hit identification?
Thank you for the question. It depends on the plan. In general, for material preparation, it takes 2 or 3 months. For assay development, it takes 2 months. And for HTS, it takes 4 months. Totally, it takes 9 months. And regarding the post-HTS, including the synthesis of related compounds, it takes 4 months.
Okay, thank you very much. No, you can provide only 9 months to the promising compound. Oh, very amazing. Okay, next question is about our capability of assay development and material preparation. Can you accept requests of assay development and material preparation as well as HTS campaign?
Yes, we can. We can prepare for materials such as purified recombinant proteins and stable transformant expressing the target protein. Using these materials, we efficiently develop assay system and conduct HTS campaign. Of course, if you have an assay system or materials, we can conduct HTS campaign using assays, client's assay system and materials after technology transfer.
Okay, thank you so much. And the next question is about hit rate. You mentioned in your presentation that the hit rate in high-throughput screening campaign is very high, more than 90%. I believe that many audiences were interested in these amazing results. Please talk about the reason of successful results of your HTS campaign.
Mainly, there are two reasons for this success. First, we have high quality and diverse libraries. 6% compounds were originally designed and synthesized. In addition, we have purchased compounds from more than 180 selected vendors. Secondly, we can offer optimal strategy depending on the target. For example, for difficult targets such as protein-protein interaction targets, we use PPI forecast library. In addition, we can conduct a virtual screen based on the information about crystal structure or chemical compounds. So the high quality library and optimal strategy are main reason for this success.
Okay, thank you so much. 90% is very, very great. Okay. Next question. I believe that the hit validation studies after high-throughput screening campaign is very, very important to get a promising hit compound. Can you conduct a hit validation study after the HTS campaign? And how do you validate hit compounds?
Yes, we can. Hit validation strategy depends on the target class or customer needs. In general, selectivity test or mode of action analysis, for example, substrate competition assay are conducted to prioritize the hit chemotypes. To confirm if hit compounds can bind to the target protein, we use biophysics such as ASMS, SPR, and so on. So we can offer appropriate plan for hit validation.
Okay, thank you so much. And the next question about the protein degradation research, especially your molecular glue library. Let us know about the molecular glue library.
Our molecular glue library was recently collected from all libraries. It consists of 6,000 compounds. It's based on the information about the patents and the literatures. In addition, we originally conducted HTS campaign for E3 ligases. Hit compounds are included in this library. So it is useful for discovery of protein degradation inducers such as molecular glues.
Okay, thank you so much. And the next question is RNA targeted drugs. How do you select the primary asset to find RNA targeted drugs?
It is important to select appropriate assay for HTS campaign. It depends on the target information. If there are solid information about the ligandable pockets on RNA, we recommend to use cell-free assay. If there are information about the RNA-BP and that is functional, we develop an assay system to monitor the interaction between RNA-BP and RNA. If there are no information about RNA-BP that is functional, we recommend to use ASMS binding assay. If there are no information about ligandable pockets on RNA or for splicing modulator, we recommend to use cell-based assay such as reportogen and artificial system. We make a flexible plan depending on the target information and the customer's needs.
Okay, thank you so much. I'm afraid we are running out of time. The next question is the final one. Final question is about IP of hit compounds. Please let us know about IP of the hit compounds and the exclusivity of the hit compound.
clients can use all the information about heat compounds, including chemical structures, non-exclusively. And it means that we don't remove the heat compounds from our library. Exclusivity period is 1 hour after completing this campaign. So we will check that there are no conflicts about the target before accepting its order. In general, it is difficult to apply for a patent of heat compound itself, but customers can optimize the heat compound to novel compounds and then apply for a patent. So IP of newly synthesized compounds will belong to the customers.
Exclusivity period is 1 year.
1 year.
1 hour.
1 year.
1 year. Okay. Okay. It's time to close the seminar. Thank you for participating in the Axcelead Global Seminar. I hope you enjoyed the learn about our company and HTS capability and service. Finally, I would like to ask you to fill the survey of the seminar. You can find the survey boxes on the web after closing the seminar. I really appreciate you taking time and joining us today. Thank you so much. Have a nice day.